Tuberculosis, TB, caused by the bacterium Mycobacterium tuberculosis, stands as one of the most formidable threats to global health in the modern era. With an annual death toll exceeding 1 million people, TB has earned the grim distinction of being the world's second deadliest infectious disease. However, this devastating impact is not an inherent characteristic of the bacterium but rather the result of a complex evolutionary journey. Recent scientific investigations have shed light on the transformation of tuberculosis from a less virulent ancestor into the highly persistent and lethal pathogen we face today. The story of TB's evolution is one of adaptation and survival. Researchers have embarked on a fascinating journey through the genetic history of tuberculosis, comparing its current form with its ancestral strains. This comparative analysis has revealed crucial insights into the mechanisms that have allowed TB to become such a pervasive and challenging disease to combat. One of the key findings in this evolutionary detective work involves the bacterium's interaction with the host immune system, particularly its response to nitric oxide. Nitric oxide is a potent weapon in the arsenal of our immune cells, typically employed to eliminate invading bacteria. This molecule is highly reactive and, under normal circumstances, can penetrate bacterial cells, causing significant damage to their DNA and other vital cellular components. However, tuberculosis has developed sophisticated defense mechanisms against this immune assault. Through meticulous experimentation, scientists identified the strains of TB's genetic ancestors that exhibited the greatest longevity in mouse models. These persistent strains shared a common feature: they were remarkably resistant to the effects of nitric oxide. This resistance was not a chance occurrence but the result of specific genetic mutations that conferred a significant survival advantage. The research revealed two critical mutations in the genes of tuberculosis that play a pivotal role in its enhanced survival. These mutations affect the genes responsible for regulating the transport of substances across the bacterial cell membrane. In essence, these genetic changes have fortified the bacterium's cellular defenses, creating a more impenetrable barrier against hostile molecules like nitric oxide. This evolutionary adaptation has profound implications for the bacterium's interaction with the host immune system. The modified cell membrane makes it exceptionally difficult for immune cells to effectively target and eliminate tuberculosis. As a result, the bacterium can persist within the host for extended periods, evading immune detection and clearance. This persistence is a hallmark of TB infection and explains why the disease can remain dormant for years before reactivating under favorable conditions. The ability of tuberculosis to withstand the onslaught of nitric oxide and other immune defenses has far-reaching consequences for public health. It elucidates why our natural immune responses are often insufficient to combat TB effectively. The bacterium's enhanced survival mechanisms allow it to establish long-term infections, making treatment challenging and contributing to the spread of the disease. However, this understanding of TB's evolutionary path is not just an academic exercise. It opens up new avenues for potential treatments and interventions. By identifying the specific genetic mutations that confer nitric oxide resistance, researchers have pinpointed potential targets for novel therapeutic approaches. Future drug development efforts could focus on circumventing or disrupting these protective mechanisms, potentially restoring the effectiveness of our natural immune defenses against TB. Moreover, this research underscores the importance of considering bacterial evolution in our approach to infectious diseases. The adaptability of pathogens like tuberculosis highlights the need for dynamic and multifaceted strategies in disease control and prevention. It also emphasizes the importance of ongoing surveillance and research to stay ahead of evolving bacterial threats. The evolution of tuberculosis serves as a sobering reminder of the continuous arms race between pathogens and their hosts. As bacteria develop new survival strategies, our understanding and approaches to treatment must evolve in tandem. This necessitates sustained investment in scientific research, drug development, and public health initiatives to combat TB effectively. The insights gained from studying TB's evolutionary journey also have broader implications for our understanding of other infectious diseases. Many pathogens undergo similar processes of adaptation and evolution, and the lessons learned from TB could inform strategies for addressing other persistent infections. In conclusion, the story of tuberculosis's evolution from a less deadly ancestor to a global health threat is a testament to the power of natural selection and the complexity of host-pathogen interactions. It highlights the remarkable adaptability of bacteria and the challenges we face in combating infectious diseases. By unraveling the genetic and molecular basis of TB's deadly persistence, scientists have not only deepened our understanding of this ancient scourge but also paved the way for innovative approaches to its treatment and prevention. As we continue to grapple with TB and other evolving pathogens, this knowledge will be crucial in developing more effective strategies to protect global health and save millions of lives.